Welcome to Let's Talk Micro. Hello, my dear audience. Welcome to another episode of Let's Talk Micro. As always, I hope you had a great week. And of course, you can always find Let's Talk Micro on Apple Podcasts, Spotify, Amazon Music, Goodpods, wherever you listen to podcasts, you can find Let's Talk Micro. As far as social media, I am on Instagram, TikTok, and YouTube as Let's Talk Micro, on X as Let's Talk Micro 1, on LinkedIn as Luis Plaza, and I have an email address which is letstalkmicro@outlook.com. So either via social media or via email, you can always reach out with any questions, any feedback, any topic suggestions. Those are always welcome and appreciated. So please go ahead and subscribe to the podcast, download episodes, and if the app allows you to do so, please go ahead and leave a review. As always, I am so grateful for your support, and as you know, the Let's Talk Micro podcast does not have any sponsors, and sometimes, I want to go to conferences and so I can get more content for you. So if you want to support the podcast in any way, you can do so via Venmo or via Buy Me a Coffee, KOFI, both links available on the show notes. And of course, if you're looking to learn more about antibiotics, please check out www.learnantibiotics.com and the Learn Antibiotics book, available on Amazon. These resources include cheat sheets, practice tests, games, and more, and they are being used by thousands of people worldwide and may be helpful for you or your colleagues. And these are some great resources from Dr. Timothy Gauthier, a very passionate pharmacist. And if you haven't checked out the previous episode, please go ahead and do so. It was a great episode about a joint infections panel, an evaluation about this panel, that this is from the BioFire system, and those of you that work in the lab, you're familiar with it, especially if you work in microbiology. At some point, either you have heard of it or you actually work with it. So in the episode, I am joined by Angelica Moran from the University of Chicago and Jaxali Arellano. She's from Rush University. And they both joined the podcast to talk about the study they did on this panel, and they talk about the procedures, they talk about the results, they talk about how they perform compared to the culture. All in all, it was a great episode, so definitely check it out if you haven't already. And before I go over today's episode, I just want to keep you updated on some things that are happening right now. As you know, those of you that have listened for a while, I am right now almost done with my master's in microbiology. And as you know, I do the podcast, I work full time, and I teach. So this coming months, October, November, they are going to be a little challenging as the end of school nears, because I have some deadlines that are approaching and I need to really commit some time to them. So all I'm letting you know is I always publish weekly, so maybe in this couple months, this coming months, you might see some weeks where maybe you have some older episodes replaying, or there might be a week or two where there is not an episode. And I truly apologize for that, but I do have to commit to school, and there's still a lot of great content in Let's Talk Micro, so if you're a faithful listener and you see that the episode, you have listened to it already, then at that point in time, maybe decide, do I want to listen to it again? Or at least, share it with someone. And if there's not an episode, I will definitely let you know as well, so stay tuned. Okay, so today's episode is about Neisseria gonorrhoeae. More specifically, about a microbicide that was developed that has activity against Neisseria gonorrhoeae. Those of you that work in the lab, those of you that know about Neisseria gonorrhoeae, right? You know that it's pathogenic. You know that you see antimicrobial resistance. So you might see some health alerts out there warning us about some resistant strains of Neisseria gonorrhoeae. So this is definitely a concern. So in the episode, I am joined by Dr. María Rendón, and she is from the University of Arizona, and she came to the podcast to talk about this microbicide. And she starts the episode by telling us about how this got started, and she references a paper where a question was asked initially, which was what would happen if commensal and pathogenic Neisseria were grown together, and it was noticed that something was happening when they were growing together, that Neisseria gonorrhoeae was being inhibited. So they decided to pursue this. And in the episode, she talks about the process, what they did to construct this microbicide. She talks about methylation signatures, how they are different between Neisseria gonorrhoeae and Neisseria elongata. All in all, it was a great episode. I imagine the potential of this, if this really has a lot of activity, how how targeting Neisseria gonorrhoeae and killing it, how beneficial this would be. And, this is a very serious pathogen. So definitely a great episode. And I said this in the episode, but definitely, it's very nice to hear about research and sometimes, to step outside the clinical lab a little bit and hear about what's going on. And so, as always, I really enjoy doing this type of talks and chatting with people and learning about this type of content. So, this is a great episode, very informative. So, let's go ahead and listen to it. So, those of you that work in the lab, you're definitely familiar with Neisseria gonorrhoeae. We work with cultures in the lab. We do PCR testing. And I saw this very interesting study that was published, and I thought it would be a good idea to talk about it in the podcast. And the article, is titled "Zap the Clap with DNA: A Novel Microbicide for Preventing Neisseria Gonorrhoeae Infection." This was published in August of this year in the Antimicrobial Agents and Chemotherapy Journal of the American Society for Microbiology. So with me today, I have Dr. María Rendón. Dr. Rendón, welcome to Let's Talk Micro.
Thank you. Thank you for the invitation.
My pleasure. So for the audience, let's go ahead and, tell the audience your name. Maybe you can start with a quick introduction, telling them what you do and anything else that you want to share with them.
Sure. I am a research scientist working at the Bio5 Institute at the University of Arizona. I was born and raised in Mexico, but moved several years ago here to the United States to get my PhD. My research interests are bacterial pathogenesis, commensalism, as well as gene regulation. And for a number of years, I worked in Dr. Maggie's laboratory, and this study was done in her lab. But Maggie retired over a year ago. So even though she retired about a year ago, I'm still working in the Neisseria field, and I'm still going to continue working in this microbicide development.
Excellent. So welcome again. So let's start with some basics here. So what was the purpose of the study?
So the purpose of the study was to develop a microbicide. But to talk really about the microbicide, first I need to describe a previous paper from the SOLAR that was the basis for this work. And this paper was published in 2019 in Cell Host Microbes. And I'm going to try to summarize really briefly the findings. So the project started with a very simple question. And the question was, what would happen if commensal and pathogenic Neisseria are grown together? So Dustin Higashi, tech in the lab, noticed that the pathogen did not grow when Neisseria elongata was present. So we were wondering what was happening there. So one of the things that we always were questioning was nutrients. Did Neisseria elongata depleted the nutrients? Or was there any toxin, antitoxin system that will account for this killing? So many years later, many experiments later, visiting scientists found that actually the DNA was involved in killing. But up to this point, we were still thinking maybe it's a toxin or antitoxin system. So finally, a PhD student in the lab, Wonjong Kim, he figured out that it was the DNA, the actual DNA from the commensal Neisseria that was killing the pathogen.
Well, that's so interesting. And I know that as I was reading, right, so I saw that there's something about the methylation signatures of Neisseria elongata and Neisseria gonorrhoeae being different. So how does that affect the Neisseria gonorrhoeae?
Yes. Okay, so I need to explain a little bit more about how this killing is happening. So on the surface of the bacteria, they have a structure known as a typhorpilus. This organelle allows the bacteria to, one of the things that it does is allows the bacteria to attach to cells. It helps them to auto-aggregate, but also it helps them to take up DNA. And they take up the DNA through a protein that is incorporated into that fiber called COMP. So COMP binds to the DNA, to specific sequences. And this pillow has the ability to retract. So when it retracts, meaning that it goes back inside the bacteria, sometimes it can take the DNA inside. So once the DNA goes into the cytoplasm, protein called REK binds to the DNA and brings the DNA into the chromosome and starts searching for regions of homology. Once it finds the regions of homology, there is going to be a protein double-stranded DNA pair where the pathogen chromosome, one strand of the pathogen chromosome is, or the two strands of the pathogen chromosome are present but separated, and then the new strand that is going to be exchanged from the commensal is present. So this in this process, the DNA of the commensal gets exchanged, but this piece of DNA will have a different methylation signature compared to the pathogen. And you were asking what's the methylation difference that they have. So it turns out that Neisseria elongata has fewer restriction methylation systems compared to the pathogen. So elongata has about, depending the strain, will have between 5 to 8 RM systems, while Neisseria gonorrhoeae, in average, they have 19. And not only the number is a difference, but also the base that they methylate. Neisseria elongata prefers to methylate adenines, and Neisseria gonorrhoeae prefers to methylate cytosines. So going back to the model. So this newly exchanged DNA, because it has a different methylation signature, the restriction enzymes will recognize it as it doesn't belong there. So they will cleave. So we believe that there are multiple sites where the DNA had recombined, and there are going to be multiple nicks along the chromosome that cannot be repaired. So because they cannot be repaired, the chromosome integrity is going to be compromised, and this will lead to cell death.
This is so interesting, right? I think sometimes, especially for me that I work in a clinical setting, and it's mostly about you work with the cultures, you do susceptibilities, but hearing you talk about all this research and the background and everything, it's so interesting. It's just it was interesting when I read it, and just listening to you talk about it, it's also definitely very interesting. So let's go ahead and talk about the study a little bit. And I know for the audience, I will put the link to this study and also the first paper that Dr. Rendón was referencing. That way you can read them at your own leisure, at your own pace, and get more information. Here in the episode, it's a more summarized. So let's go ahead and talk about the study. Maybe you can start with the microbicide design, and then we'll go from there.
Sure. So based on what I previously said, we thought that can we use this to our advantage? So we know that bacteria, they are pileated, and they would like to take up any DNA that is present as long as it has a DUS sequence. And for the killing to happen, we know that the methylation signature should be different. So if we will pass our DNA through E. coli, the methylation signature will be completely different. So mostly will be adenine methylated, or they can also methylate cytosines, but it's not going to be the same recognition sequences that the restriction enzymes from gonorrhea has. So we decided to PCR amplify regions of Neisseria gonorrhoeae chromosome and clone them into a vector. And we went with Neisseria gonorrhoeae because we did not want to affect commensals. So there were 3 types of sequences that we selected. The first ones were genes that are not essential for in vitro growth and that are rarely found in commensal Neisseria, like TDFF and IgA. They have some function for the bacteria, but for our purpose, we just wanted to see if we can kill the bacteria using these sequences. Then the next set of sequences were DNA methyl transferases that are essential for Neisseria gonorrhoeae. And we chose 3 of them because they have been identified that are active in a variety of Neisseria gonorrhoeae strains. Those are NGOA1, NGOA14, and NGOA15. And also, they are not commonly found in commensals. And then the third set of sequences are essential genes, even though they are also present in commensals. So each of these sequences was PCR amplified, and we did not include any regulatory region, like any promoter or stop codons or start codons. So because we didn't want anything to be inserted and by chance into a commensal, and then now they can produce the protein. So we PCR amplified each of the sequences and cloned in a vector. But these sequences will have were flanked by the DUS sequence, so the bacteria would be able to take it up. So we built about 12 different constructs, and in some 9 of those had single targets, and some of them were concatenated, meaning that 2 or more targets were present in one construct. So each of the plasmid was, once it was built, was purified and tested in a killing assay on the laboratory adapted strain Neisseria. So all these microbicides kill Neisseria gonorrhoeae with different efficiencies, but the ones that target essential genes work the best. So one of those, MB14115, was selected for testing with antibiotic-resistant Neisseria gonorrhoeae and with low passage isolates. And in all the cases, it worked really well.
Okay, so it worked really well. And then were there any effects on commensal organisms?
No, the commensals were not affected. So the DUS that we selected to use there, so Neisseria has different DUSs. The central part, the DUS10, is conserved among the Neisseria, but we selected the DUS12 that is mainly used by Neisseria gonorrhoeae. Some of the commensals have a DUS12 that has one of the bases is different, and it does make a little bit of a difference. But no, the commensals that we tested did not get killed.
Well, this sounds like very promising, like it has a lot of potential. And I know that you mentioned in the article that there's as a potential delivery method, you talked about a lubricant. Maybe you can talk a little bit more about that.
Yes, we decided to use, so we needed a way to deliver the microbicide. So an easy way is personal lubricants. It's going to go to the genital area, and for the lubricants to be sold, the FDA requires all the lubricants that are sold and marketed in the US to have a clearance. So we thought that it will make it easier in the future when we are ready to, or if somebody wants to move it forward.
Okay, and with any experiment, right, or any study, you always get some things that maybe work well, some don't, you get some limitations. What worked well in the study and what didn't, if there anything?
So I think like everybody else, what this study, part of this study started at the beginning of COVID. So that, as anybody else in the world, it made it a little bit challenging. Sometimes because supplies were a little bit difficult to get or will get more expensive. And at the beginning, the universities closed, but we were lucky that the University of Arizona allows some people to keep working, especially because my work has to be, I have to be in the lab, in the wet lab. I could do some things at home, but most of what I need to do is in the lab. But luckily, we were able to get a permit to continue working, but everything was slower. There were other experiments that we would have wanted to do that had to be canceled, again, because of COVID, and it was with some collaborators that unfortunately we could not do. That was part of the challenging that was out of my hands. Concatenating also the sequences. In one case, I could not get the concatenated sequences. But I was also running short on time to finish this paper. So I didn't try harder, just for time, not for willingness. And something, what worked well, the microbicides really work, some of them work beautifully. They killed 99% of the bacteria in the assays. So that was nice to see. And the other thing that we haven't talked about here is that one of the 3 of these targets that we had are essential genes in Neisseria. Of those, GRA and PAR-C are known to have point mutations that will make the bacteria resistant to antibiotics. So we were careful to have sequences that were sensitive to the antimicrobial. Because we also wanted to know, are all the strains getting killed, or some of them are taking the DNA and recombining it? Because there was a point mutation, especially with when we tested the antibiotic-resistant Neisseria, we could monitor whether or not they had taken of the DNA and recombined it by just looking at that point mutation. So very few bacteria were able to take it and recombine, because we found that now a mutation, especially that happened with a PAR-C. They took the sensitive allele and recombined it to its chromosome. And when we measured the antibiotic resistance, now it recovers some sensitivity. So one of the things that in the future we would like to see, if we could also use this microbicide to restore antibiotic sensitivity in Neisseria.
Wow, that will, yeah, if that works, that will be definitely great, right? I mean, we definitely with all the, especially all the reports that we hear about, resistance increasing in Neisseria gonorrhoeae, and so that will definitely be great. And this microbicide, if it definitely sounds like very promising, and when you mentioned, right, definitely in COVID was challenging time, and for many factors, right, and especially when it comes to things like that, your work that you need to be in person, and then at that point in time, a lot of things were shifting towards being remote, and the supplies, that's a pain that we all felt in the lab. So I definitely hear you. So you talk about this, and maybe what are some other next steps for this?
So we would like, so I would like to work more in the antibiotic resistance, and also work more on this microbicide, because I think there are some sequences that we could add that will improve the uptake, the DNA uptake. So those are the things that I will be testing in the future. And that is in the application side of the microbicide, but also the basic science. So what is really happening? Because in some cases, we are wondering if the bacteria is trying to go through a replication, and the replication is halted because there are some nicks. So we want to look at the basic science behind that. Also, some of the bacteria got killed way more efficiently than others. So we also would like to understand why is that happening, because that could inform us in a way to modify the microbicide to make it more effective.
Okay, well, definitely, if I always pay attention to things that have been published out there, but know that as you progress with your research, and you're definitely more than welcome to come back and update the audience or talk more about this. Definitely, you're more than welcome to do that. Before the episode comes to an end, is there anything else that you want to add or tell the audience?
No, I hope that people enjoy the work, and we will keep working on this.
This was great, and especially for those of us that work more on the clinical side, and it was kind of like refreshing, and just listening to a little bit of research. And so definitely thank you for taking the time to being in Let's Talk Micro.
No, thank you for having me.
My pleasure. And that, my dear audience, it's the end of this episode. I hope you enjoy learning about this microbicide for Neisseria gonorrhoeae. As always, I enjoy sharing this information with you. So please continue bringing that passion to what you do. It's so important. You do such great work. Thank you for your support. Thank you for your understanding as this couple of months approach. As I said before, that there might be some weeks where there might be an older episode playing, or there might not be an episode. Thank you so much. Once we get through this, I will continue bringing that great content that you always enjoy. So, as always, stay motivated, stay safe, and of course, continue talking micro. Until the next time. Bye.